Welcome to our Allied platform. Last week I introduced excretion and we looked at the various organs in the body which are involved in the removal of metabolic wastes from the body. We looked at the kidney, which is responsible for filtration of blood and removal of wastes like urea, excess water, and also salts. We listed some of the functions of the kidney. For example, it is an organ for excretion of nitrogenous substances from the body, regulation of water content of the body fluids, regulation of pH. Also, it maintains the concentration of mineral ions and some other substances. And lastly, it is also involved in the production of red blood cells. We also described the functional unit of the kidney, the part of the kidney which performs all those functions and is known as the nephron. There is this part of the nephron. It is found within the cortex. Then this region of the nephron is found within the medulla. From here we can see that this is the cortex of the kidney, this is the medulla of the kidney. Therefore, the cortex of the kidney, we have the proximal convoluted tubule, the distal convoluted tubule, the Bowman's capsule, the afferent arteriole and efferent arteriole. However, in the medulla of the kidney, that's where we usually have the long loop of Henle, which is mainly used in reabsorption of water. We ended by looking at the two types of nephrons found within the kidney. There is the cortical nephron, which is mainly found in the cortex, and a small part of it, that is the loop of Henle, goes into the medulla. The juxtamedullary nephron has a longer loop of Henle, which is found in the medulla. That's why this one is mainly used in conditions when there is less water in the body. We shall see that the longer the loop of Henle, the more water which is selectively reabsorbed into the body. Our lesson today, we are going to discuss urine formation, and also that we shall also look at the loop of Henle has a countercurrent multiplier. Urine is formed by mainly three processes. One is known as ultrafiltration. We shall see that blood is filtered under high pressure, and small substances like water, glucose, urea, and some ions pass through the Bowman's capsule. Then we shall also look at selective reabsorption, where all substances which are still useful to the body are selectively reabsorbed back into the blood. And lastly, tubular secretion. That is when the tubes of the kidney, they also add on more substances. For example, as we say that the kidney is involved in regulation of pH. During the process, some substances like hydrogen ions may be added onto the filtrate, and this is known as tubular secretion. Let us start with the ultrafiltration. Ultrafiltration takes place in the Bowman's capsule. As you can see from the drawing, you find that the afferent arteriole is larger than the efferent arteriole. Therefore, there is more blood which comes into the Bowman's capsule, and there is less blood which is drained from the Bowman's capsule. There is always excess blood which remains in the glomerulus. This one leads to an increase in hydrostatic pressure, which causes filtration of blood. You find that only small substances like glucose, water, urea, ions, amino acids, vitamins, they can pass through the walls of the Bowman's capsule. However, all large substances like cells, for example, red blood cells, white blood cells, proteins, they do not pass through the Bowman's capsule. However, sometimes the efferent arteriole may decrease in size, may further decrease in size, that is vasoconstriction, which leads to excess pressure in the glomerulus. You may find that some large substances may also be able to pass through the Bowman's capsule, but this is not common. The next process is selective reabsorption. Whatever is useful to the body, like glucose, amino acids, most of the water, are selectively reabsorbed, and this takes place in the proximal convoluted. We have seen ultrafiltration taking place somewhere here. Then in the proximal, we are going to see selective reabsorption of useful substances to the body. In the drawing is the section through the proximal convoluted tubule. This is the lumen of the tubule. Then this one forms the wall of the tubule. However, it is also surrounded by blood vessels known as the vasa recta. In this drawing, blood vessels are not represented, but this efferent arteriole surrounds the proximal convoluted tubule, then the loop of Henle, then the distal convoluted tubule, and also the collecting duct. However, for simplicity, it is not shown, but the efferent arteriole surrounds the tubule and is known as the vasa recta. What happens is that substances which are still useful to the body, for example glucose, diffuses from the lumen into the surrounding cells. Water moves by osmosis from the lumen into the surrounding cell. In the case of some proteins, as I told you that in some cases the efferent arteriole may further constrict, which increases pressure in the glomerulus and may lead to filtration of some of the proteins. What happens that those proteins, suppose they have been filtered and they are part of the filtrate, they are absorbed somewhere here at the base of the microvilli, and they form a pinocytic vacuole. Then it fuses with a rhizosome and digestion takes place. The amino acids are used by the cell. Then glucose and some other ions from this cell which makes up the tubule, they are actively transported from this cell into this space. This space is occupied by tissue fluid. Tissue fluid occupies the basal channels. However, glucose ions, they are actively transported from the cell which makes up the proximal convoluted tubule into the tissue fluid. From the spaces between the tubule cell and the blood vessel, movement occurs by diffusion and also some other processes. This is because of the difference in concentration gradient. You may find that you may have more water in this region but less water in the blood vessel which surrounds the proximal convoluted tubule. Because of the difference in concentration gradient, substances from this region, they just move by simple processes which do not involve the use of energy. And the last process is tubular secretion. As I told you that we are going to look at regulation of blood pH by the kidney. In the process, some substances like hydrogen ions may be further added onto the filtrate. Then down the loop of Henle in the descending limb, we are going to see that water is selectively reabsorbed. However, in the distal convoluted tubule and also the collecting ducts, For them, they are mainly involved in regulation of water, meaning that selective reabsorption of water in the distal convoluted tubule and also collecting ducts depends on the amount of water in blood. If there is less water in blood, then there would be more water which is selectively reabsorbed in the distal and collecting duct. However, when there is enough water in blood, no selective reabsorption of water in the distal convoluted tubule and also collecting duct. Now, how does the loop of Henle concentrate the urine such that it is more hypertonic compared to the blood? There is active secretion of ions in the ascending limb of the loop of Henle into the medulla. As you can see in the drawing, this arrow represents sodium ions which are being pumped actively from the filtrate across the cell into the medulla. This concentrates the medulla with a lot of ions. Therefore, this one will cause movement of water. These black arrows, they represent movement of water from the filtrate through the cells of the tubule into the medulla by osmosis. Therefore, the ascending limb of the loop of Henle is permeable to water, and water continuously moves out by osmosis. This is because of the increased amount of salts which are actively pumped from the filtrate by the ascending limb of the loop of Henle. Therefore, as the filtrate descends down the tubule, more water is selectively reabsorbed by osmosis. Because more water moves out of the filtrate by osmosis, the concentration of the filtrate increases, as you can see, from 300, 600, 1000. Therefore, concentration increases until it reaches the hairpin bend, where we have the highest concentration of the filtrate. Then, when the filtrate ascends the loop of Henle, as we have seen that ions are actively pumped out of the tubule, and this one requires energy. The concentration of salts and ions continues to decrease because of their active secretion into the medulla. Also, the presence of salts and some ions in the medulla causes water to be reabsorbed by osmosis, because in this region, the medulla, there is a lot of ions. Therefore, water is selectively reabsorbed, and also this side is selectively reabsorbed. Therefore, by the time urine moves out of the collecting duct into the pelvis and also the ureter, it is highly concentrated. And in this way, the loop of Henle works as a countercurrent multiplier. It is countercurrent because you can see that the fluids move in opposite direction, and also it's a multiplier because the concentration of the filtrate increases down the loop of Henle, and this one causes reabsorption of more water back into the blood. As we have seen that this tube is surrounded by blood vessels. When water reaches into the space between the tubule cell and also the blood vessel, it moves into the blood vessel, that is the vasorector, by osmosis. What happens is that in the collecting duct and also sometimes the distal convoluted tubule, the permeability of those tubules to water depends on the effect of hormones, as we are going to see in osmoregulation. In the table, we are seeing the percentage concentration of substances in blood plasma and also urine. In urine, there are no proteins, as we have seen that they are large particles. Therefore, because they are of high relative molecular mass, they cannot pass through the Bowman's capsule. Glucose is selectively reabsorbed. It passes through the Bowman's capsule, however, it is selectively reabsorbed back into the blood. Urea, its percentage concentration increases. As we have seen in the countercurrent multiplier mechanism, more water is selectively reabsorbed in the loop of Henle. Because of that, the percentage concentration of most of the substances will go on increasing, as you can see in the table. Also urea, you can see that urea is not selectively reabsorbed in most cases. And there is also tubular secretion, whereby some of these substances are secreted and also added on to the filtrate, which also increases their percentage concentration in the urine. And lastly, adaptations of the distal convoluted tubule. With adaptations, we know very well, we look at the structure and how the structure is related to perform its functions. Also, the gene adaptations of the loop of Henle or the nephron, that is the relationship between structure and the function of the nephron, as we have already described the structure of the nephron and also the kidney, and you know very well how it functions. You can relate the function, the structure, and also with the function. I wish you the best. For any question, you can contact me. In our next lesson, we are going to look at other functions of the kidney, that is osmoregulation. How does the kidney maintain a constant amount of water in the body? Also, we shall also discuss the RAS system, where the kidney maintains the concentration of sodium ions in the body.